Hello everyone, let's do year 2009, the main paper. Now, first question would be quite simple for you. You're asked to label the different parts of the digestive system. So the esophagus, make sure you spell O-esophagus, esophagus properly. Pancreas, which is a leaf-like looking organ, is connecting to the duodenum and the small intestines. Briefly describe two functions of the liver. Now, given you a mind map over here, you can either talk about blood glucose concentration regulation, or you can talk about other things. So I would choose either this or this if you want to elaborate. Now, some of the keywords that I'm looking out for in regulating blood glucose would be the fact that you mentioned the hormones, insulin and glucagon, and that the glucose concentration is kept 40 to 90 milligrams per 100 cubic centimeters is optional. For deamination, we're most concerned with removing the amino group because that's the part that's going to convert to toxic ammonia, but is eventually converted to urea, which is something less toxic, and is removed through the urine via excretion. So the carbon part, which is carboxyl part of the amino acid, can be converted to glucose and used again. And we know that excess glucose is stored as glycogen. You can also talk about other functions such as detoxification, whereby you mention the enzyme name of alcohol dehydrogenase. Or you can talk about the recycling of the iron when red blood cell is broken down by the spleen. Hemoglobin is recycled. Or you can talk about synthesis of protein. The simplest would be that you can also talk about the liver making bile, which is important for emulsifying fats. The definition of emulsification would be at the bottom over here. Okay, let's take a look at part C, whereby digestion is aided by three different types of enzymes. If we're looking at big groups, we are looking at carbohydrases, proteases, and lipases. If you want to be specific, let's talk about the different stages where starch can be broken down into maltose, eventually to glucose, for proteins to polypeptides, and eventually to amino acids. And last but not least, fat globules. They are emulsified by bile, which has no enzymes, into oil droplets. And this is where, when its increased surface area to volume ratio allows lipases to act on it to eventually break fats down into fatty acids and glycerol. So question over here asks for the enzyme, substrate, and end products. Okay, next question. Scientists carries out a breeding experiment. So we're looking at test cross. He crosses all the tall plants with dwarf plants of the same type. And they told you in the question that the tall and the dwarf are all homozygous. So over here, you realize that this is where they put their big T allele, small t, assuming that the allele for tall is known as a capital T, and the allele for dwarf is small letter t. Then we move on, and they say that all F1 generations, of course, they were all tall. So what can you prove? It means that the moment you have this big T allele, and if it's dominant, you will be tall. Okay, so genotype, the moment you have a big T dominant allele in your genotype, you will be tall in terms of your phenotype. Okay, let's answer the other side, which is the dwarf part, whereby you need to mention that even in the hetero state, meaning big T, small t, okay, the dwarf is still masked, because in order for the dwarf phenotype to be expressed, you need a homozygous recessive genotype, which is small t, small t. Next part B is just a definition question. We are looking at the definition of genotype, in which is the genetic makeup of the organism. Phenotype is what is expressed and usually is a physical trait. Part C, scientists then do another cross. You cross the two heterozygous, so you do your working to write your big T, small t over here. And then when you do, you will realize that you have a 3:1 ratio of tall is to short, or dwarf. Okay, next part, last part. Now we know that the theoretical ratio is 3:1, Unfortunately, you don't have half an organism. So the ratio is 23:7, which is very near. This question is not asking for whether it's whole number or not, but more of the expected ratio as compared to the whatever that's produced may be different because we're looking at a small number. So sample size is very important. We can also talk about other things such as whether the pollen transferred to the stigma, whether there's ovum available, so on and so forth. So these are the technical part when pollination takes place before fertilization. And then you can talk about during fertilization. Each time the gametes fuse is also independent, and that's why the genotype is different. Okay, and therefore it did not meet the expected ratio. Okay, question 3. Under the chapter of impact of man on the ecosystem, we did come across using microorganism for environmental biotechnology. So in this case, this shows an activated sludge process used to treat sewage. Another one over here, which is just the wastewater treatment process. I want you to take note of the similarity. So over here, in terms of sequence, there's always wastewater or dirty water, so to speak. There's a settling tank, which you can talk about. And then whatever that settles to the bottom is the sludge. Now, if you're looking at using aeration for the bacterial, it means that the bacteria used for the first step is aerobic bacterial. Okay, this that the bacteria thrives in oxygen, air with oxygen. Okay, and all this wastewater can actually be channeled to a settling tank where the bacteria is removed. Whatever that you treat can be discharged back into the sea. For sludge, you actually need anaerobic digesters. So this question can be linked to the next about aerobic respiration. Okay, so in this case, aerobic respiration, we're looking at glucose with oxygen, carbon dioxide and water, and a lot of energy is involved. In one, you can put large amount of energy over here. Okay, why air is pumped, as mentioned, if it's aerobic bacteria, then you need oxygen for respiration, and as it respires, as it grows, it will feed on the nutrients in the sewage. This also increases the surface area to volume ratio of the sewage in contact with oxygen, and the rate of diffusion increases. Rate of bacterial activity also increases to act on the sewage. Okay, let's do part B. Now, this is a scenario whereby untreated sewage was actually released into the river. I want to bring to the concept that we learned about eutrophication again. So this is a simplified one. If you're interested, you can look at this one over here. Basically, the idea that nutrients are leaching into the water body. So you can mention things like nitrates and phosphates, which are all good for growth, especially for algae and water plants. Submerged plants will die if they overgrow because they cover the surface, and submerged plants don't get the sunlight. Then bacteria grow rapidly. As they grow, bacteria use oxygen. Okay, and this, they will decompose dead plants. And in return, as they decompose the dead plants, they grow more, they use more oxygen. So other organisms will also die due to the lack of oxygen. So these are the steps needed, or the key phrases needed when you're explaining eutrophication. Question 4, factual question. You should know by now where's the trachea, the bronchiolae, and the alveoli. Structure to function, I've written some over here. There are 4 main ones that you can talk about. Now, I want you to think about what you would rank in terms of priority, or what is the so-called more important answer. So I've highlighted some here for you. Right, one cell thick epithelium, thin film moisture, highly vascularized, spherical in shape to increase surface area. Now it's always structure to function in biology. So take note, always link back to your question, and feel free to pause the video to look at the 4. Part C is a factual question. 3 major toxic components in cigarette or tobacco: nicotine, tar, and carbon monoxide. So you need to know the 2 specific example and how it caused. Elaborate on it. Question 5 is relatively simple. Match the name of the blood component to its function. Because when you're asked to write an open-ended answer, please be more specific than this. Okay. Red blood cell contain more hemoglobin if they have no nuclei. That's the reason. So more space for hemoglobin, so that it can bind reversibly to oxygen and transport it from the lungs to all over the body. Why veins have valves, you can talk about 2 parts: prevent backflow, and that the direction will now be in one direction. Capillary walls, why 1 cell thick, we're looking at a shorter distance for faster diffusion, and therefore exchange of materials, nutrients, and gases. Coronary heart diseases due to occlusion, which means the blockage. Okay, to be specific, if cholesterol is deposited, then we talk about atherosclerosis. Some of the causes can be diet high in fat, stress, and smoking. Okay, question 6. Question 6 actually compares 2 kinds of plants, the cactus versus the buttercup, and it told you that one is in hotter or drier parts, the other one may be in tropical. Both cactus and buttercup have stomata. So this is a graph measuring the size, how big it is, versus the time of the day. It's drawn 2 lines here for you. Always compare what. So over here, buttercup seems to be low overall in general, whereas for the cactus it decreases till noon is so-called the hottest, and therefore it remains size remains relatively small, versus the buttercup, which is super high. Okay, then the last part where buttercup goes low again, the cactus size of stomata opening increases also. Okay, what are some parts? Now just compare whether you have a day, midnight, the day part, and then the noon part, and then you compare. So there's 3 portions to compare. Then give the reason. So I always describe the graph first. Describe graph, and then give my explanation. In this place, the graph, there's 3 different portions, so that's what I'm going to do. Let's try to analyze the answer. So they talk about cactus plants, where they are found, why is the stomata closed. You want to prevent or reduce the rate of transpiration to prevent excessive water loss. Okay, at night, it opens to allow carbon dioxide and so-called water, but let's be concerned with the gas, to diffuse in the plant so that glucose can be synthesized by photosynthesis. In contrast, so there are words of comparison, plants are found in an environment with normal temperature and humidity. So in the day, stomata of the buttercup open. Okay, so this is normal for the other plant. Okay, although the rate of transpiration is high, the good thing, or most likely the buttercup will have enough water to replace whatever that was lost, compared to the cactus, which is in the desert. So at night, it closes again. So this is the normal condition. So this is some way to organize your answer. Describe the graph, then give the explanation. 3. Make sure you have words of comparison. Alright, this is the next part. Each root hair cell, long thin protrusion, okay, or elongated portion, we can use that too. There's also extension of the cytoplasm, so there are quite a few, basically to increase surface area to volume ratio, if you're talking about the elongated portion. Okay, then you can talk about it containing a lot of mitochondria to release energy for active transport, because sometimes mineral uptake or even water uptake can be against the concentration gradient. Okay, so the third one you can talk about is the cytoplasm being extended. Question 7 asks the shape of the pyramid. In this case, the first one is biomass and the second one is number. We know there's a so-called inverted relationship here. So let's see how answer can be answered. So a pyramid of biomass, upright in shape, versus the other one, inverted, 3 having the smallest number because there's only one 3 and the rest is upright. Correct? Okay. Then you can compare why. So you can have some explanation about the bottom building blocks obtained through fleeting. And then you can talk about why it decreases at each stage, because biomass available to the next traffic level may be lost in terms of energy. Okay, that's how you describe and compare. Part B is actually very specific. They ask for photosynthesis and energy transfer. Light energy from the sun is trapped, so it talks about how it enters the whole food chain, and then talk about how it's transferred and why energy is lost. So these are some things to take note of. Question 8. Now we need to remember the interconversion between glucose and glycogen. Hormone insulin converts excess glucose to glycogen. When we don't have enough glucose, glucagon, right? So glycogen is converted back to glucose when the hormone glucagon is secreted. Okay? So all this happens at the liver in terms of its conversion. So let's take a look at this graph here. You realize that, okay, glucose peaks before insulin. Okay, so this makes sense because the change in glucose concentration is detected by the hypothalamus, which actually sends signals for the pituitary gland to secrete insulin into the blood, and eventually the liver is the one that converts glucose to glycogen. Okay, so using the information, describe and explain the production and function. So this cascade or the mechanism is as such. Tell me why glucose increase. Okay, because after a meal, digestion, absorption. Okay, so once the blood glucose is detected, okay, pancreas, the hypothalamus actually asks in the islets of Langerhans to secrete. So this part you need to take note of. So it should be hypothalamus. Okay, and then the pituitary gland will actually ask the pancreas or send signals to the pancreas to secrete the insulin into the blood. This will correspond to the increase in insulin and therefore the conversion of glucose to glycogen. And you can even elaborate more about how, in what ways glucose can be converted or decreased. Okay, we always have the negative feedback mechanism, and since it's a glucose regulation, when it drops below the norm, what happens? The whole thing repeats itself, correct? Okay, I'm gonna actually give you an image to help you remember all this. There we go. Okay. So, let's see how this is done. So basically, when blood glucose level is too high, what is being stimulated? So detection, right? Stimulus, the rise of blood glucose is the stimulus. Receptor is actually in the hypothalamus, which picks up the increase in blood glucose concentration. Then it stimulates the pituitary gland, which stimulates the pancreas to release the insulin into the blood. Okay, and this whole event repeats. What happens? Body takes up more glucose. Glucose also converted into glycogen. Okay, read up more about what the body does to decrease glucose concentration. Okay, when this is too low, what happens? Okay, stimulus declining, and this side repeats itself. And that's all we have.